Vampire bats can, when food is low, eat food, regurgitate food up for a partner or a friend, and they will do that. So this article is trying to investigate how and why those relationships are constructed. They're mostly focused on the mechanism and how do you get to such a high cost behavior, because if you share food with someone and then you can't find food the next day, can't find someone to share with you, you die. Bats have about a 2 to 3 day window where if they don't find food, they won't survive. So the first thing I look at in an article, and a good place to start, is a summary or an abstract. And here I'm always looking for, what are they trying to prove? Why are they conducting the study? And what did they accomplish in the study? Because usually they have a main conclusion or takeaway from the article there. And this is important for you to start understanding the article, and it will usually guide you through the article and talk about main conclusions they made in the sort of order that they do them. And it's also important, if you're looking for research to use for a paper or something, for you to determine quickly if it's relevant or not. So these are the two most important sentences I found in the summary. So their theory that suggests an individual can reduce the risk of exploitation by initially spreading out smaller cooperative investments over time. So that's going to be their grooming behaviors. And then they said their main conclusion is that we found compelling evidence that vampire bats selectively escalate low-cost grooming behavior before developing to high-cost food-sharing relationships. Some discussions. I always like to read all the headings first. And I always like to keep in mind that heading throughout the results, because sometimes the results can get really technical. They can include a lot of methods, which we'll talk about in a second. It can be really confusing and hard to read. So if you keep that main heading in mind, it's usually the conclusion that they've drawn from that bulk section of results. Articles often, but don't always, have some sort of brief description of how they came to that conclusion within that results section. So if they said, our conclusion is new food sharing relationships were more likely when introduced bats lacked familiar partners, how did they test that? How did they know that the bats that they had were not familiar with each other? How did they prepare and set up and introduce groups? How did they induce food sharing? Because what they did was fasted the bats. Are either described completely before the results or introduced before the results, or sometimes they're not mentioned at all, and they're only at the end of the paper. They'll say, we use this technique to do this thing, and they won't describe the method very much until a detailed section later. In this paper, the methods are listed in complete detail at the end of the paper. So this article actually chose to introduce and illustrate their methods, which I think is really interesting. They didn't do it for all of their methods, but this one is really clear. This is because when you read that paragraph that I just showed you, this one, it's kind of this huge group of, okay, we took bats from here and here, and we threw them together in these groups and this group. But here they're showing you exactly how they did it. So they had groups from the two different areas, and they separated them off, and they had them sitting by themselves. They controlled and introduced them in different size groups, and then they kind of jumbled them all together. The important part about this too is that they're proving to you, they're showing you empirically that the bats that they took, very, very low chance of being related. And they discuss that more in the statistics section, which I skipped over because I'm not qualified to teach you guys about biostats. This article does have a really detailed on their statistics if you're interested in that. Every lab that I've been in and talked to has had their own specific way that they like to run statistics, their own programs they like to use, their own methods and statistical analysis that they like to use. The important thing is that when they say something is statistically significant, or they use the word significant, they mean something that is statistically significant. So it has some way, and they define that significance somewhere in the paper. This paper, and I didn't realize this until I had already chosen to do this, they don't explicitly state their hypothesis, which I wish they would have. which is a broad statement meant to describe their overall observation. So since they stated their conclusion in their summary, they kind of skipped over, instead of doing an introduction, they skipped over the part where they said, this is what we think is happening and this is what we're trying to test. Predictions, and there's usually quite a few of them in an article, are saying, if that hypothesis is true, how do we expect our experimental group or the model system to respond to those different situations or experiments? So each heading under the results section in this article, where it has that conclusion, is a response to a prediction. I think this is the first or second heading. It's not the second heading. New food sharing was rare and emerged after social grooming. If vampire bats follow a raising the stakes model for food sharing, then steps must be taken to form a relationship. Since food sharing is a high-cost behavior, it should occur infrequently. So this is basically saying, if the raising the stakes model is true, and that is how they are forming these relationships, then we have to build up to that, because that's the point of raising the stakes. And if we're raising the stakes, and the point of raising the stakes is that sometimes we'll find cheaters and it won't work out and we won't continue that relationship, food sharing is going to be less frequent than the lower cost behaviors. So that is the predictions that they're addressing in this heading. And then you see those results and you can go through and figure out how they came to that conclusion based on how they first decided to test that prediction, and then how the results from that test supported this conclusion based off of this prediction. So it's kind of following the scientific method of form a hypothesis, form a prediction that can be tested to either support or negate the hypothesis, and then follow through. I don't know if these are usually that active, but if anyone wants to try and make a prediction based on this, I would love to have some participation. If anyone wants to give it a go.
I feel like I'm cheating because I read the summary, but I think there was something about them starting off with grooming, and then slowly, as they sort of like reciprocated the grooming, going on to food sharing more. I don't know if that would count as a prediction.
Well, that's exactly what they're testing in the paper. They believe that grooming leads to it. So in this case, the word predicted is the really key word. If we're saying that grooming leads to the higher cost behavior of food sharing, then we should expect that the bats will start with grooming dynamics, and that bats that don't groom won't continue on to create food sharing, but bats that do groom are much more likely to lead to food sharing. And to say that if you see grooming behavior and grooming dynamics, then you're more likely to be able to conclude that in the future a food sharing relationship could form. So that's definitely on the right area.
So if you were seeing bats that were food sharing but hadn't groomed before, would that be then sort of negating that prediction?
Exactly. That's exactly what would happen. If you saw bats start food sharing before they ever had grooming, or I think there's also another behavior, but they don't talk about it very much, that's huddling for warmth. If you saw food sharing before those other two things, that would negate the prediction. Negate your prediction, you subsequently negate your hypothesis.
Okay, that makes sense. Because they're taking that risk and they're not protecting against cheating.
Exactly. That's exactly what they're trying to figure out. And they're trying to figure out, because food sharing is a really high cost and risky behavior for them. They could die if they can't make up that food loss that they shared with someone else. So that's exactly what they're looking for. This is another thing I realized that probably made this not the best example. They don't have a specific conclusion section. Their conclusion is labeled evidence for the raising the stakes model, but it is their conclusions. So this is where they summarize their results, and they also talk about supporting evidence from other papers. Supporting evidence from other papers could also be included in the introduction, but they didn't really have a set apart introduction. They more had a summary. So they also talk about future studies and work that needs to be done. One thing that stuck out to me in that section was that this is a lab experiment, and we know we've seen over and over again that animals don't always behave in the wild the same way they do when we put them in the lab and under observation. So the authors talked about how similar experiments need to be done, but out in the open. They also discuss alternative hypotheses and why their results and their experiments fit the raising the stakes model better than some of the other models. And they also discuss how their work can be applied to other topics. So does anyone have any questions about that kind of general workflow through the article? Also put out the two figures because I figured that would be a good thing to talk through. These figures are definitely stylized, so they're not the kind of graphs that I'm used to seeing in molecular biology that are very strict black and white bar graphs. They're meant to show different things, so I thought you guys might want to talk about the figures. So this is demonstrating, obviously the heading is the first place to look in what they're doing, and you always want to look at your axes. So this axis on the y-axis applies to all three graphs, A, B, and C. Probability of being groomed in a trial before the first food donation increased. And I don't understand exactly why the month mattered, other than to show that it took months for these relationships to form. Like, it was not something that happened quickly.
Wait, so what does grooming mean here?
Grooming is a behavior that the bats do where they pick relatively low cost to the person doing it, but it benefits the person receiving it.
Oh, sorry, your voice cut out in between. Could you repeat that?
I'm sorry. Grooming is a behavior that's pretty low cost. When you groom someone else, it doesn't hurt you, but it benefits them. It doesn't benefit you to groom someone else, except that they'll then groom you in return. So the idea here is that they're watching for relationships that go back and forth that are cooperative.
All right, thanks.
Here they're watching how long it takes for those grooming relationships to form and when a food sharing relationship can form.
What are the three different graphs showing? Are they showing different bats or different? I'm not quite sure what they are.
I'm going to be totally honest, I'm trying to figure that out.
Okay, that makes me feel slightly better.
What's the gray? Oh, sorry. What's the gray part mean? Because I don't understand the gray part.
I am working on it.
Oh, sorry.
I'll be totally honest, I wrote 2 papers on this paper, and I did not use this graph. The main point of the graph here is that they're saying that sharing escalates. So I'm sorry, increasing grooming escalates. So you start with doing it not that often, and then you increase in rate. And we'll talk about that in the next figure more, because it only increases up to a certain point. So this is actually, I think it's demonstrating probability. It is. So probability of being groomed in a trial before the first food donation. These are 3 different trials. Basically, they're looking at 3 different groups of bats. Relationship formed in the dashed lines, and a food sharing relationship did form in the blue lines. So they're showing the growing rate. I assume that gray area is the error, which could either be standard error of the mean, standard deviation, something like that. This is a very stylized version of a graph. In typical graphs, you could see error bars, which I think would be much more clear. That would be a criticism I have for this author. Yeah. This graph, it's basically saying in the dashed lines, no food sharing relationship occurred, and they're showing that grooming didn't escalate between those times. So if no food sharing relationship occurred, there was much less escalation in grooming. Whereas when a food sharing relationship did occur, which is the solid line, we saw a huge increase in the probability of a grooming relationship.
I'm not sure if I've sorry, I'm not sure if I've interpreted this right, but is B for bats that didn't know each other before, and A, bats that did know each other before? There's something. Sorry.
That's correct.
Okay.
So that's 3 different trials, and in each trial they varied relationships with captive-born bats. B is, so A, this is right here. Can you guys see my mouse? A was detected in previously unfamiliar bats. B was in relationship with captive-born bats. So that would be the couple of babies that were born during the trial. And
Okay.
C, divergent grooming.
Is there a reason why A and C there's quite a big difference between where feeding relationships do occur and don't? So there's solid and dashed, whereas they're a lot closer together for B. Is there a significance to that?
I assume that that's because B is involving captive-born bats, which are going to behave a lot differently than bats that were born in the wild and brought into captivity in life.
Okay, that makes sense. Thank you.
Because those are the babies that have been had with the colony and have been with the same group of bats their entire life, whereas these bats are being brought together and introduced.
Right. Okay. Thank you.
This is the last figure, and I actually like this one a lot. So they're demonstrating that grooming behavior up until food donation increases a lot. But once you get to that first incidence of food donation, grooming behavior sort of stabilizes to a certain point. So they don't increase anymore. Like I said, there's a lot of statistics in here, and those things are very important to a research article. P, I will tell you, P is equal to 0.005 is incredibly unlikely to be due to random chance. But depending on the journal, depending on the discipline, that p-value could be anywhere from 0.5 to like much more zeros in order to demonstrate statistical significance. It depends on a number of different factors, both in the experimental design and in the field, of what you need to achieve. Oh, sorry. Do that. So this graph is a lot easier to see. So this is where the first food donation occurs, is at 0. And then, as you can see, it did take them a really, really long time to hit that first incidence of food donation. Easier to read, in my opinion. It makes more sense. It's obvious that we're talking about leading up to food sharing. The lines are both grooming relationships, but the solid line is before we have our first food sharing, and then the dashed line is afterwards. So afterwards, we sort of reach a relatively stable probability of having a grooming incident in the bats. About this graph? Yeah, that graph is a lot clearer. I think this one makes way more sense. Sometimes it is not great to share one axis with everything. I don't love the way they did that other one. So that's all I have for the main article itself. I did a Times article, and I think it's really interesting to talk about science in the media. And I'm sure you guys will talk about that at some point in classes, and how we represent science in the media. So I just wanted to throw up some food for thought, because I think this is this is an example of where the media represented this article incredibly well. I think it's accessible. I think it lacks on talking about the methods and how they did that pairing, and how they proved that the bats didn't know each other, and how they controlled for the experiment. But I think it's really accessible for people to read, and especially right now, we're seeing so much misinterpretation of scientific research in the media. So I just think that's a really interesting question. of how we can make science accessible. And then I have to show you my citations because I don't want to get caught on copyright. Do you guys have any questions? Is there anything else about the article that you want to talk about? Does anyone have any biology questions that I can answer? That anyone needs homework help or anything like that? Because I've got some time, so I can always. Glad I can pull up a whiteboard here too.
Sorry, are you like helping with homework? No.
I can do, yeah.
I missed a bit of the stream, I don't know.
Sorry?
You're helping with homework.
I can help with homework. That's what I have for this article, unless anybody has any more questions or wants to talk about a specific part of it.
So, could you talked about like how like you would see other graphs in like normal research articles. Could you like show like an example of those graphs, like bar charts, the ones you said?
Maybe. Let me pull up one of. Can I stop sharing my screen for a second?
Sure.
There. Let me find an article really quick. I will show you guys what I have while I look for this. Somebody asked if I've enjoyed having a wider foundation in biology. Yes, I don't. So I'm studying molecular biology right now, but I intend to go into forensics in graduate school, which is a sort of application of biology. So it's really helpful for me to have a sort of general idea of different fields of biology and a different way to do it. It's also just a lot of fun, and it makes it easier to communicate in it. Where is it? Of articles, and I can't find any of them. I think it makes it easier to sort of talk to people. In research, I specialize. I study something called islet cell biology. So this research article I used for my classes, but it's not my research area. I specialize because most of the professors you're gonna find that are doing research are specialized. So it's you guys are gonna get to see it if I show you some graphs here. Okay, that's not a bar chart, but it is a specific graph. I ended my stream, I'm sorry. I was just going to do that when I stopped sharing my screen. Let me show you guys this. So this is from an article called Conformational Prerequisites for Formation of Amyloid Fibrils in Histones. The specifics of understanding what amyloid fibrils are not the pancreas. It is not like the islets of Langerhans. They're the cells that produce the chemicals that are involved in glucose regulations. It has to be type 2 diabetes, whoever said type 1 diabetes. So it's interesting. It's incredibly specific. I study amyloid proteins here. Like this, think the kind of thing that causes Alzheimer's plaques, prion diseases, that sort of thing. You don't really need, I'm not going to be able to walk you exactly through that. What I can show you is that I think it's a lot clearer in talking about, these are emission spectra, so they took readings at each of these every single point of what they did. Oh, CD spectra. I really want to learn CD. That's circular dichromacy. It's a way to measure conformation of molecules, stuff like that. So it's very biochemical. See, you can tell very clearly that they took a measurement at each of these different wavelengths along these bars and were able to measure rotation. That's an angle, that's theta. So that is an example where you can look at this and you can look at that and you know exactly what they're looking at, as opposed to having to sort of sort through the axes and if that gray box, which it was error, which I think it was on it, which it should. Maybe it shouldn't. It's a really specific. It is a really specific machine. It's sort of like NMR or something like that. I think it's a lot easier to read. Like, does this make sense? Where you can really clearly see where each data point was taken. I stopped sharing my screen. Maybe I can find a bar chart. A whole book downloaded on here. Oh, here's some error bars. Let's look at some error bars. Here you go, guys. This is what I was talking about, where everyone see that with the they took a data point and this looks like genetics. I hope it's emission again. Sorry. I haven't read this article in a really long time, but this is the kind of thing I wanted to show you guys, where molecular graphs tend to be a lot less stylized, a lot more focused on putting a data point and giving you the error. So this is the data point here in the middle, and these are the error bars surrounding it. And here, these yellow ones have really tiny error bars.
So it's like standard deviation.
Could be. It could. More frequently, I see standard error of the mean. And I'm sure it says somewhere. If it doesn't say somewhere in this box, it will say in the methods how they plotted it. I might not even have the methods saved. Sometimes methods are a completely separate file, especially in molecular, because you get these methods that are a page long. So they'll upload them as a separate file, and you can read the article without reading them. There's also some electron micrographs if anyone's interested in that. And they're looking at these different sections. That's what an amyloid fibril looks like, or can look like. That's not the one I studied. Actually, it is the one that I studied, or it's very similar to. But that's one way that they can form these little fibrils, and you don't want those, they're bad. But you're going to see more specific graphs that are less stylized and more to the point, and also more clearly labeled on the graph. Like you can see here, they've labeled the exact thing that they're looking at without insulin. It's plus insulin, minus insulin. I don't think it says here whether it's standard error of the mean, standard deviation. There confidence intervals you can also use. I haven't actually used a confidence interval since I got to college, but I did learn how to do them in high school. So there's a couple different ways you can measure error. Like I said, it tends to be pretty lab and or journal specific. Standard deviation is going to give you usually the widest, and standard error of the mean kind of shrinks that error margin down to smaller. In my experience, that's not always the case.
Yep.
Anything else you guys want to look at? See if I can answer any of these questions more specifically. Like I said, most research is gonna be a lot more specialized. And there's you can get so specific. You can get just study one molecule and one cell type in one disease, apply to a much broader field, and I learn about that field, and that's why I have all these articles like on bovine amyloid proteins. But whoops, don't want to show you that. Whoops. Actually, I don't think there's anything there, but I think that's all I have. I'm waiting to see if there's any more questions in the chat, or I can do like I said. If I hear no objections, I think that's it. But I hope that was helpful, at least fun. I like talking about the bats. I think that's a good article. I hope everyone has a great day. Bye.
Thank you for this.
Of course. Have a great day.
Thank you. You too.
And don't forget to donate to the donate thingy. I think he posted the link. Sidespin posted the link somewhere. Donate to their charity for the free streams. Have a great day, everyone.